Hello, everyone, and welcome to today's live broadcast, Modernization of Animal Studies, presented by our three presenters, Natalie Bratcher, a 3Rs scientific liaison and animal welfare specialist at Abbvie, Laura Schaevitz, Vice President of Scientific Technologies at Vium, and Sean Maguire, a GSK Associate Fellow and a Comparative Translational Sciences Veterinarian at GlaxoSmithKline. I'm Susie Valdez, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational and virtual events and webinars. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on the Ask a Question box located on the far left of your screen. Type that question into the drop-down box that appears on the screen. We'll answer as many questions as we have time for today. If you have any trouble seeing or hearing this presentation, click on the Help Desk button located on the promotional board at the bottom center of your screen, or use that Ask a Question to let us know you're having a problem. This presentation is educational and thus offers continuing educational credits. Please click on the accreditation button located on the promotional board and follow the process to obtain those credits. Following the presentation, if you have any questions by our audience members, they will be answered via email by our presenters. Please join me now in welcoming our three presenters, beginning with Natalie Bratcher. I will now turn the presentation over to her. Welcome, Natalie.
Thank you. Thank you so much, Susie. And thank you. I'd really like to first thank LabRoots for asking me to be a part of this program. It's an honor. I believe this is a really valuable topic that merits attention, and so I'm excited to be a part of this panel and share the hour with my co-presenters. So bear with me here as I get the hang of this. All right, so Abbvie has a strong commitment to responsible research. We're really committed in various ways, and we have various groups that support this commitment and to ensure due diligence. As a part of my role, I facilitate and drive 3Rs for impact across the company globally, or at least that's my goal. We do this a number of ways: through our Global Alternatives Committee initiatives, through various research initiatives aimed at 3Rs impact and improved welfare, and through our involvement in 3Rs consortia, external 3Rs consortia. So also important to note is through our 3Rs Advancement Award program, we often identify areas of 3Rs impact across the global company, which is how I actually became interested in exploring novel endpoints, modernization of endpoints, and these new digital vivarium technologies. So important, an important I think aspect of this, of all 3Rs research, is collaboration. I am going to cover this much more in my introduction here, but I feel like 3Rs really have more impact when we collaborate and work together across the industry or even within your own institution. Before moving on, I'd like to acknowledge some of my colleagues. So Steve England here at Abbvie, who actually gave a presentation at LabRoots last year on this similar topic and how he used some digital vivarium technologies here at Abbvie. But I'd like to thank him for sharing some of his expertise and knowledge with me. I'd like to thank Dr. Letty Medina for her leadership and support. I would like to also thank Dr. Sheppenbaron for his leadership in the 3Rs and for sharing some of these introductory slides with me. We gave this presentation at another venue, and he kind of served as the moderator and introduced the topics, and so he shared his slides with me. So thank you, Stepan, and for all that you do for the 3Rs here in the U.S. And then I would like to acknowledge my fellow panelists presenting with me today. Finally, I'd like to acknowledge both the North American 3Rs Collaborative and the IQ 3Rs Leadership Group. Probably more than acknowledge, just bring attention to the fact that they exist. So if you're unfamiliar with either group, I encourage you to visit the web links here on this slide to learn more and to get involved. So this is the standard schematic of the R&D process. It makes a number of assumptions. It assumes we have a fixed rate of attrition. It suggests that we're locked into R&D, strict R&D timelines, and that this preclinical process is 6 to 8 years. However, we often need to be challenging these timelines. This also assumes that because of the amount of time and cost required for clinical development, that this is where we would make the most improvements and efficiencies. However, if finding innovative ways to enable improved decision-making earlier in the drug discovery process, so over to the left of the image, if doing so requires challenging the timelines, then it may be necessary, as better decisions in early drug discovery could reduce attrition rates and increase success in clinical development. So this is definitely a model that we need to challenge. So the images here were taken from a Nature Reviews drug discovery publication. Now the publication is 5 years old now, so these figures may be slightly different, or they would look slightly different if we reevaluated these numbers today. However, the figure on the left breaks down failure by cause, with efficacy being the leading cause, and the figure on the right breaks down failure by therapeutic area. So we need to continue evaluating and reevaluating how we approach R&D within pharma for success. We have to challenge ourselves to be more innovative and to challenge the status quo. Perhaps one of the reasons for attrition is that we've been too reductionist in our approach. Regardless, across industry, we have not been as successful as we would like or could be. So applying approaches such as systems biology, engaging microphysiologic systems, advancing translational sciences, and integrating new technologies may enable us to make better decisions, leading to fewer failures. So these are just a number of technologies available aimed at identifying endpoints or increasing the value of added in vivo modeling. So many of these technologies kind of have different endpoints or have different value, depending on the therapeutic area in which you're using them and depending on what they were developed for. And these are just some of the technologies available that are working their way into the preclinical in vivo process across discovery. Now, I'd like to take a moment here to thank these companies for allowing me to use their logos, but I'd also like to acknowledge that there's probably many more that are not shown here. So if you are actually a part of one of those companies, I apologize that we missed you, and please reach out to us and let us know who you are and what your technology does. We're really excited to explore some of these. So we are engaging novel technology, which is an evolving approach, and in doing so, we actually could be having a number of benefits. So we could be enabling cycle time reduction, improving reproducibility and or decreasing variability, and ultimately improve translational relevance. This is also generally where we will have 3 R's impact either directly or indirectly. So this slide here kind of covers various aspects of what we need to evaluate in these technologies for value. So, some of these technologies generate massive, see if I can get it to run. Yeah, massive data sets, which is great because this is increasing our capability of data analysis and interpretation, enabling data storage and the capability to retrospectively review data. Most of these technologies require assessment and validation before they can be comfortably adopted into an R&D toolbox or toolkit. Some of these systems are not yet scalable. For some, that's an issue. For others, they don't need to be scalable. So that's a factor that should be considered. Cost is obviously a factor with any new technologies. Most of them are rather costly. However, this is where cost-benefit analyses come in and are often required because we may find that some of these technologies actually enable us to do more with less, or that the value added will justify the cost or justify the means. We believe the best way to evaluate such technologies is through effective collaboration and benchmarking. Oops, let me go back. So The one way that we're doing this is through the IQ Consortium. Bear with me one moment. Sorry, I'm working between two screens. So the Innovation and Quality Consortium is a not-for-profit organization of pharma and biotech companies with the mission of advancing science and technology to augment the capacity of member companies to develop transformational solutions that benefit patients, regulators, and the broader R&D community. Within the IQ, there are a number of leadership groups, one being the 3Rs leadership group, which has multiple goals and working groups to help drive these goals for impact. So basically supporting decision-making through efficient group learning and to direct guidance and evolution of the technology, improving its value to develop, to both the developer and the customer. So providing feedback to these technologies and the companies that create these technologies to help them evolve is critical. It's, you know, it's kind of like they provide these technologies to us, we validate them and evaluate them and provide them with the feedback to better the technology and better the capabilities. So one thing that we are working on, and this is very, very new this year, is a digital vivarium scoping working group with the goal of sharing strategic interests, challenges, and value propositions for digital vivarium technologies in the pre-competitive space. So sharing non-proprietary experiences, data, and ideas regarding digital vivarium technologies. And then we're also evaluating the current state of digital vivarium technologies. So doing this collaboratively, we are hoping will be much more effective. So there's many values to being a part of a collaboration and approaching something using a collaborative effort. One being that we're able to optimize information collection and share this information in the pre-competitive space. There are many benefits to synergizing efforts, but I think the most valuable is that they allow us to broadly increase the reach of impact of these initiatives. Engagement and collaborative approaches allows us to leverage collective input to establish a global strategy and support our internal strategies as individual companies. In alignment with this, we can develop consistent messaging and guidance rather than each company kind of figuring it out on their own. And finally, with regard to new technologies, collaboration enables us to collectively develop expectations and testing strategies. There we go. So this slide is a fun one. It's one that I actually borrowed from a colleague. And when I saw it, I was really intrigued. And I felt like it was an impactful enough slide to show again because he put forth some real thought-provoking observations. So, you know, coming back to the R&D process, which I talked about a bit ago, we are increasingly aware that disease biology is very complex. With few exceptions, we know that if we aim at a single target, they often don't translate into favorable clinical outcomes. Also, with exceptions outside of infectious diseases, there are very few drugs that are disease-modifying or alter the underlying cause of a disease. The majority of our drugs are aimed at treating symptoms of disease up until now. Now, there is a potential space, target space, that's viewed as undruggable by small molecule and large molecule approaches. Our increased knowledge of basic science is not translating to innovative or impacting innovation or impacting how we approach drug discovery and development. As the cost of R&D is increasing, there are still very few new game-changing therapeutic modalities. So medicine has not really changed in many, many years, right? However, as we learn more about the genome, the approach will hopefully change in medicine to one that is aimed at prevention rather than treatment or cure, and Big Pharma's approach will have to adapt accordingly. Currently, however, pharma's approach has not changed. So how is this relevant to new technologies? So, give me a moment to get back to the schematic. So this, again, is the schematic of the preclinical process. So the left side of the schematic I showed you earlier. So the question is, how do we apply these new technologies in this process for the most impact? So while the in vivo models are in the right place here on the schematic, they're over to the right, right? So we're doing a number of things prior to putting our compounds into the in vivo animal models. So that's great. And with in silico modeling, improving, and we're doing a lot of really unique modeling, which is helping to inform these in vivo studies. So we're on the right path. However, once we get to this area here on the right with in vivo studies, how do we reduce the number of animals used here by improving approaches to the left, or perhaps new technologies? How do we improve the value of the in vivo models here on the right so that those moving forward, those compounds moving forward, continue moving forward successfully rather than dying once they get into the clinic or clinical space? So perhaps applying some of these new technologies will enable more value in the in vivo studies and help to improve decision making when it comes to selecting the appropriate compounds to move forward. That's the hope. So this is a study, I thought it was worth showing. This was a publication in Regulatory Toxin Pharmacology demonstrating that CNS effects account for 10% of drugs withdrawn. So these side effects such as dizziness, nauseousness, which are quite difficult to detect in rodents, in the rodent models, we're missing those. And so we need to assess how these new technologies and new endpoints might enable detection of subtle CNS effects or adverse events earlier in the drug discovery and development process as well. So it's not just discovery. If you remember the reasons for failure graphics that I showed a bit earlier, there, I think it was 26%. For PCS in terms of reason for failure. So they were right behind efficacy for safety was. So, so as we assess these technologies, there's going to be a ton of benefits, but there are also a number of limitations that we need to evaluate because it's completely relevant in whether or not they will be useful for us for various disease models. So one example of limitation is ensuring optimal housing for rodents. So let's say you're phenotyping a specific strain of mice. Are you getting an appropriate phenotype if they're single housed? And so some of these systems don't have the capability to track animals as a group yet. And so that's something we would like to probably assess and look at when we're comparing the various technologies. Currently, there are some systems that enable social housing. However, the capabilities and experimental endpoints of each system are different. So the ability to individually track an animal might be challenging for one system using one set of endpoints, whereby another might have overcome this limitation, but their endpoints might be different. Another example would be nesting, and another consideration, I think an important consideration. So based on some published research, for optimal welfare, we should be providing 6 to 8 grams of the shredded paper nesting material shown here in the image. And thanks, Brianna, for sharing this fun image with me. So if you're not familiar with Brianna Gaskill and Joe Garner's research, I would suggest that you look into it. I know they're both speakers here as a part of the Lab Animal Sciences Conference, but Brianna's done a great deal of research looking at appropriate nesting for optimal thermal regulation in mice. And so some of these systems actually would not allow for that optimal nesting amount, because if a mouse builds this complex nest, which is what we want it to build, and is in the nest, and let's say our endpoint that this digital technology is measuring is a breathing metric, how can you monitor breathing if the animal's hidden in their nesting, right? So it's going to be a give and take, and so these are the things we need to assess. Another consideration is, are we providing the animals enough in their environment to accommodate for convective heat loss if they're in an IVC, for instance? So scalability is something I already discussed, but I think it's important. Depending on your need, scalability might be important, it might not. Another limitation worth considering is obtaining molecular readouts in continuous readings without disrupting the animals. So one of the main benefits of some of these systems is the hands-off approach and not being in the room. However, some of these molecular readouts and continuous readings at this point in time require some type of intervention. And so working with these technologies and these companies to try to help us come up with the ability to get these readouts without disrupting the animal. So finally, just kind of going through summarizing the benefits of the new technologies, both from a scientific perspective and a 3R's perspective. So first, these technologies can enable earlier decision making, as we'll be getting improved endpoints. New endpoints or improved endpoints. Novel endpoints are always important and worth pursuing. Because we certainly need to look for new opportunities to improve our process, our scientific process. Now, I will comment here that this isn't always necessarily going to be an either-or approach. These technologies may not replace standard endpoints or standard models, but rather just be value added. These technologies may enable us to use fewer animals as we obtain more data from each subject. So that would be a great opportunity for reduction in a 3R's context. We may find improved reproducibility and decreased variability with continuous monitoring of animals, leading to a decreased need to repeat studies. By removing the component of the experimenter, again, having the experimenter in the room changes the animal's behavior so much. And there's plenty of data to show this. So we may actually see less variability in the data and a truer picture of what's going on with the animal when the investigator is not in the room. And we'll assume also, if that's the case, that the animals will be less stressed, because we do have data showing that even just having somebody walk in the room, you see a change in behavior. With integrated historical data, we may be able to use fewer controls based on standard conditions. And from an animal welfare perspective, we all know that death is not a justified endpoint. And from a scientific perspective, we lose a significant amount of valuable data if an animal dies unexpectedly. Say a rodent in a cancer model, if they die unexpectedly or we're not able to get to that animal before they pass and collect our valuable physiologic endpoints, we've lost valuable data. So some of these technologies actually enable earlier intervention. They will alarm you, and they have basically endpoint determinations. And so this would allow you to go in and intervene, get the animal, put them down humanely, get the endpoints that you need, and therefore leading to less pain, distress, and death. So finally, continuous video monitoring allows the capability for data mining. So to be able to go back and retrospectively look at your data, review animal behavior, look for potential factors influencing the animals, this actually may lead us to novel endpoints or discoveries. So with that said, I am going to pass the presentation over to my co-presenters. I'm really excited for them to share their presentations with you. One of them is on one of these amazing technologies. Laura Schaevitz will be presenting the Vium technology, and then Sean Maguire will be presenting how they've used some of these technologies at GSK. So I look forward to answering any questions you may have by email. Don't hesitate to reach out. Thank you so much for your time. And with that, I will close.
Thank you, Natalie, for that introduction. And thank you for the opportunity to let me tell you a little bit more about how we're leveraging automated data collection technologies to enhance study reproducibility and animal welfare in preclinical in vivo research. Just to give you a little sense for what I will be talking about today, first I'll introduce Vium's continuous smart cage monitoring system. And then I'll talk a little bit in my brief time about how we're using this system to improve scientific practice, as well as to look at various aspects of animal welfare. First, a short introduction on the Vium Smart Housing platform. So here we've built a platform that's used to monitor animals in their home cage in real time. The system's built for scale. So if you take a look at the left, you can see our mouse racking system in which we have 49 cage positions that are continuously monitoring animals at all times. The real workhorse of the rack is the slab. If you turn your attention to the center of the slide, you can see a white box which is above one of our standard Abbvie cages. In that box, we have a camera as well as other sensors, including illumination, temperature, and humidity sensors, which allow us to monitor both the animal's environment as well as information about the animals 24/7. All the information from that slab actually gets sent up to the cloud, and I'll tell you a little bit more about that in a second. But in essence, all of that information goes through the little box which is shown in front of the Vium rack. We use standard Abbvie cages, which just slide in and out of the rack with no effort at all, no connections. And inside we've built proprietary Vium cage furniture. So you can see potentially on that lower image there a running wheel, as well as we have a ladder and a food hopper. And the cage furniture is built in such a way that there is no location in the cage in which the animal can hide from the camera. So we get a fairly accurate view of the animal and its activities 24/7. We also use the cage furniture to learn a little bit more about the animal. So for example, we have the ability to monitor the amount of time that the animal spends running on the wheel, and there we can learn more about the circadian rhythms as well as some of the other metrics that we have going. One other slight difference with the Vium Smart Housing System is that we also have each of these cage positions individually illuminated. So we use regular blue enriched LEDs during the daytime and IR LEDs during the nighttime. So it doesn't matter where an animal is on the rack, the animal will receive the exact same amount of illumination, whether they be on the top or the bottom or the front or the back of the rack. And that's just one way that we try to standardize the environment for all animals. If we go to the next slide, all the information from the smart housing system actually gets piped up into the Vium cloud. And in the cloud, that's where we do all of our advanced data processing, computer vision algorithms, and that's where we pull out the metrics, so motion and breathing rate, and I'll go into a little bit more detail about how we calculate those in just a moment. But then we also do analytics, so we look at group averages or changes within single animals over time. And all of that information is delivered to the researchers over the Vium Research Suite. So on the right of the graphic there, you can see a standard view of our online web portal, which you can log into through just a standard Chrome browser. And there you can see individual animal metrics and the ability to monitor the animals via video real time, as well as see their changes in motion and breathing rate, as well as wheel running time over time. So just to give you a little bit more information about how the Vium metrics work, here you can see a up-close image of an animal in the Vium cage. And overlaid are a series of green dots. And we actually use complex computer vision algorithms to monitor both speed of motion as well as breathing rate in the animals. And we compare frame by frame where each pixel in each of those green dots is. And pixels that move large distances tell us how fast the animal's moving. So if you look at the upper animal, you'll see that there are vectors pointed in the up and up to the right, and that's showing us that that animal is moving up and to the right, and that the lower animal, there are no green vectors there, so that animal is still. So again, that information is read out directly as speed of motion. And then we actually look for breathing rate, we actually chest heavals for periodic motion that are going basically in equal and opposite directions associated with those chest heavals. And that's what we read out as breathing rate. And to ensure that we were getting accurate breathing rates, we actually put animals inside of a plethysmograph inside of the cage and read out breathing rate using more of the gold standard compared to our computer vision metrics, and found that the breathing rates were highly correlated, so upwards of 0.99. Again, we also read out circadian rhythms, or wheel running, not shown on this slide. And from that, we've derived a number of circadian metrics as well. So the ability to look at entrainment, active period, and activity bouts. So how do we use this data to improve scientific study outcomes? Well, we've worked across many therapeutic areas, including respiratory diseases, autoimmune, safety, oncology, just to name a few. And really what we look for are partners who have scientific questions that they have trouble answering during in-life data collection. So I wanted to share with you today just one example of a group that came to us with the cuprizone mouse model of MS. So here the challenge was that it's a fairly good model for studying demyelination and remyelination, but it's a fairly long model. So it takes about 6 weeks to induce full demyelination in the animals, and then there's a period of 3 weeks in which you look for the ability to remyelinate. And during that time, there are no real overt signs or symptoms that the animals that demyelination is occurring. So there are some indication that by looking at rotorod or perhaps open field, you could see some changes, but they're infrequent and inconsistent. So the majority of researchers who study cuprizone may look at body weight, or most importantly, they look at histology across the course of the study. So the question was, could the Vium platform be used to monitor changes in motion and be able to see some of these subtle changes in motion that arise from demyelination? So just to give you a sense for how we ran this model, you can see on the left a timeline just showing you the overview of how we ran the model. So as in all traditional experiments, we brought the animals in and we fed them control chow for a week. And during that time, we gathered baseline data on the animals. So we looked at motion as well as breathing rate and took conventional measures such as body weight and clinical observations. And then we randomized animals such that all animals had, or all groups had equivalent average motion and breathing rate prior to putting them on cuprizone chow. So then the animals were put on 0.2% cuprizone chow for 6 weeks. And during that time, again, we monitored motion and breathing rate, as well as more conventional measures such as body weight and clinical observations. And then following that, we took the animals off of the cuprizone chow and put them back on the control chow to allow for remyelination, and took brains at a number of different time points. So the data for this model is still fairly young, but I think you'll see that there are some pretty obvious and significant changes. So just to orient you to these two graphs, if you focus on the nighttime motion to the left, you can see that we have days on the x-axis and the amount of motion on the y-axis. And that dotted line represents when the animals were actually put on the cuprizone chow. Prior to being put on the cuprizone chow, both the control chow animals, so the dark lines, as well as those on cuprizone, showed very similar motion profiles. Within 5 days after being placed on cuprizone chow, however, we see a very significant decrease in motion in those animals put on cuprizone chow. Interestingly, cage changes induced a recovery almost to baseline in those animals. And then we see, of course, a decrease in motion again. The second cage change also induced a recovery, but not all the way up to baseline. So suggesting that we are, in fact, seeing the effect of cuprizone chow. And again, we'll look to see whether or not this is supported by the histology. Interestingly, we also see a change which is mirrored in breathing rate. So again, breathing rate is very similar prior to animals being put on cuprizone chow, and then we see decreases in breathing rate before both the first and the second cage change. So having looked at this data and comparing it to literature, we see that changes in motion and breathing rate do correlate or match with what's known about changes in when demyelination starts to occur, again in that first week in animals put on cuprizone chow. So we'll look at histology to ensure that what we're seeing from the behavioral standpoint matches what we're seeing from the histological standpoint. But it is a suggestion that just being able to monitor animals in their home cage will provide a lot of valuable information about the status of the animals. What might be evident here is that just doing standard procedures on animals actually have a fairly significant impact on those animals. Therefore, we're actually spending quite a bit of time better understanding how you can use the platform to monitor animals, not only to improve study reproducibility, in essence, by understanding how procedures can affect studies, but also just looking at animal welfare more generally. So to do that, we're actually using a retrospective technique. We've collected many years worth of animal videos, and rather than running specific experiments to test some of these hypotheses, we're going back through and mining the data that we've already collected. And so some of the questions that we're starting to ask are, what is the impact of standard procedures and or the environment on animal welfare and model predictability? So to take us back to that question around cage changes, what I want to do is show you just a little bit of information that we're starting to gather around the effects of cage changing and also various different ways that animals can be marked, specifically tattoos and ear tagging. So what I'm showing you on the top of this slide here is just a standard trace from a single cage showing motion over time. The dark bars are nighttime and the light bars are, of course, daytime. And you can see a very consistent circadian pattern with increased activity during the nighttime and fairly little activity during the daytime. What I'm showing you is the day before, the day of a procedure, as well as the day after the procedure. And in this case, where that red line is on day 0 is where we gave the animals in this cage a cage change. And you can see a very significant and prolonged increase in activity associated with the cage change. Just as a comparator, now I'm showing you data traces from animals that received tail tattoos. So in this case, just a simple 1 or 2 lines marked on the tails, as well as ear tagging. And again, this is just a simple plastic clip that was put into the ear. Overall, you can see that the response of the animals for both the tattoo and the ear tag is significantly less than the cage change. To quantify that, we actually looked at cage changing in 36 pairs of C57 females, as well as looking at tattooing 61 pairs and ear tagging 117 pairs of animals. And just to give you an idea of baseline, we looked at the same animals that had received a cage change, but at a time where there were no procedures. So again, those were 36 pairs. And what's plotted here is the percent change from day -1, so the day before the procedure. Again, as you expect, a very significant increase in motion in the animals that received a cage change, which was still significantly elevated even the day after the cage change. In contrast, when we look at the tattooing, the blue line, you can see really no difference on the day of the procedure and maybe a slight decrease the day after the procedure. Ear tagging, a very similar result. There's no real change the day of the procedure and a significant decrease the day after the procedure. Interestingly, we see a similar pattern between tattooing and ear tagging versus cage changing in nighttime motion. So here, cage changing had no effect. We didn't see any change in nighttime motion the night after or 2 nights after the procedure, whereas tattooing and ear tagging both involved a decrease in nighttime activity, both the night after, or the night of, and then the night after the animals received either the tattoo or the ear tag. We were also able to look at breathing rate. And so here, we now have a dissociation between animals' responses to tattooing and to ear tagging. So again, in comparison to the day before the procedure, both cage changing, the red line, and tattooing, the blue line, we saw elevated breathing rates, both the day of the procedure and the day after the procedure. Whereas ear tagging and no procedure, there were no differences across days. So it's actually quite interesting. We've now repeated this, while I don't show you the data, on another couple of studies and found similar results in each of the studies. So in general, we see the following results, which is cage changes, a significant increase in daytime motion and in breathing rate. So likely that increase in breathing rate has something to do with the animals moving around quite a bit more. In contrast, we see slight differences in animals' responses to tattooing and to ear tagging, where both tattooing and ear tagging result in decreased motion, both day and night motion after the procedure, but only tattooing results in increased breathing rate. So as we think about what are procedures that we can do that provide the least disruption to the animals, one would think that perhaps ear tagging would provide slightly less disruption than tattooing of animals. But we're doing more work to kind of sort that out. Some of the other questions that we're looking to answer: can we improve humane endpoint determination? And can we better assess analgesic efficacy following routine surgeries? So just to give you a little sense for what we're doing around humane endpoint prediction, here's data from an oncology model in which we gave animals, we implanted glioblastoma cells directly into the striatum of animals. And actually in this study, we saw more animals found dead than was ideal. And so actually this analysis was used to go back and look to see if we could have used biometrics to better predict animals that were reaching humane endpoint. So what I'm showing you here are data from both motion and breathing rate. And unlike in previous data where I've lined data up according to study day, here data is lined up according to the time that the animal died. So 0 here is the last day that each of the animals was alive. The animals that reached study endpoint and thus were not sick are shown in the black. Animals that were found dead are in the dark blue, and animals that reached humane endpoint and then were euthanized are in the light blue. And what we've plotted here is a percent change day over day. And what you can see is that in the last couple of days, so specifically on the day before and the day of, there's a significant drop in motion which can be identified across the animals. Unlike with motion, though, we don't really see a signature in breathing rate, at least in these animals. So we went through and said, well, if we had used a 50% loss in motion over 2 nights as a humane endpoint, how successful would we have been in identifying animals that had been found dead versus animals that had reached humane endpoint versus animals that should have been maintained until study endpoint? So in the first column here, you can see the percentage of correct identifications. So again, in this study, we lost more animals than was ideal, 15. If we had used this 50% night over night loss in motion, we would have identified 10 of those 15 animals as having reached humane endpoint. Again, we would have identified 18 of the 20 that did reach humane endpoint via other criteria, such as body weight. And we would have correctly identified that 7 of 7 animals that should have made it to study endpoint would have made it to study endpoint. And just to give you a sense, and then of course there were no false negatives, or false positives. So we wouldn't have incorrectly identified that an animal had reached humane endpoint when it had not. So this is just an example of how we can start to look for patterns at the end of study in life period and be able to predict when animals might be reaching humane endpoints. We're actually in the process now. We've collected a lot more data since this study, and we've identified maybe 5 or 6 different patterns which commonly occur at end of life across a variety of different therapeutic areas. And we're in the process of looking to see if we can develop predictive analytics to identify those patterns in those animals. So these are just a few examples of some of the work that we're doing to both understand how continuous monitoring of animals can improve both the science as well as animal welfare aspects. So in terms of the science, can we actually identify, or can we use some of these metrics in order to gain better functional readouts of demyelination, in this example for the Cooperzone MS model? And I believe that my colleague Sean will tell you a little bit more about that in another model. And also, the impact of how we can use continuous monitoring of animals to improve animal welfare. Can we more identify endpoints by identifying these signatures across a variety of therapeutic areas? Can we refine procedures to minimize the impact? Can we change how we, or can we best identify the ways that we should be marking animals to provide the least disruption to those animals, as an example? So this is just some of the work that we're starting to do. Thank you so much for giving me the opportunity to tell you a bit about this. And at this stage, I will go ahead and turn this presentation over to Sean Maguire.
Thank you, Laura. So I'm going to get started on my portion of this discussion. And before I really get into it, I want to thank a former colleague of mine, Brian, He was the one who really put together the majority of this slide set, and was also really critical in the vision that GSK has in terms of looking at this technology. Brian is now at the National Toxicology Program. As was on the intro slide, my name is Sean Maguire. These are some other semi-famous Sean Maguires. I'm not any of those. And so this is the disclaimer that I like to use. The other thing that I would add is that if my voice starts to stutter, that's not the fault of the streaming technology, but I am someone who occasionally stutters. So I want to briefly go over what I'm going to be covering in the next 15 or 20 minutes. And that's just to touch on some of the criticality and the rationale behind why we use animal models in drug development, some of the challenges that are associated with their use, and where there may be some opportunities to dramatically improve their translational value. And then, most importantly, I want to spend some time talking about a proof of concept study that we did in collaboration with the very smart folks at Vium. Animal studies are used throughout the drug development process. Starting all the way, in the very beginning when we start to identify targets, all the way through to preclinical safety. And for those of you that are not familiar with drug development, there is this pyramidal issue in that, right, we start out with thousands of compounds, and as we learn more about these compounds and the biology behind what these compounds do, the number that we actually progress into the human clinic whittles down. But so why do we use animal models at all? And so at its most basic and simplest level, right, there is a great amount of conservation across species. And so a good example of that is the cardiovascular system, right? So if you look at the both the anatomy and the physiology across, say, the mouse, dog, and man, you find that it is pretty well conserved, and so it is with that idea that studying drugs or potential drugs in other species should help us understand what will happen in the human patients. Developing drugs is a long-term process. It generally takes 10 to 12 years. It is very, very costly, and so especially when you consider the cost of not just what it took a program to get from its very beginning all the way through to market, but all of those other failures that happened along the way, right? We are unfortunately not so smart that we can, at this point, predict very well, when something gets into first time in humans, whether or not it will make it to market. And so the number that's quoted here is that only about 10% of the programs that make it into the clinics actually make it to market, right? And so certainly from an animal use standpoint, there is a real challenge here, right? We want to be making sure that we use the animals on studies that will give us, real, accurate, precise, reproducible, meaningful data, because to do so otherwise would not be a proper use of the animals. And so when we look at once programs make it into the clinic, why they fail, right? 2/3 of those reasons for failure are things that we very specifically look at in our preclinical models. And so, right, said a slightly different way, this points out a translatability gap in that what we thought we knew based on the preclinical models turns out to be not fully correct. And then so these potential medicines end up failing. And so in the last 5 or so years, there have certainly been a number of papers that really question the value of the animal studies that we're doing, right? If we're doing these animal studies and we are not correctly predicting what's going to happen in the clinic, then what is the value of those studies? And there are two main concerns there, right? One is about the translational relevance, but the other is about the reproducibility, and that certainly within the same institution, right? We have seen cases where studies are repeated, but the outcomes end up being different, and so either we're not being appropriately precise about the way that we're working, or there are other factors going on that we don't have a firm understanding of. And so some of the developing technology has been very helpful and useful in trying to increase the translational relevance as well as improving on the precision and the reproducibility. And so an example of that is the use of telemetrized dogs. So this is a way, it's a very hands-off, potentially round-the-clock way of monitoring changes in the cardiovascular system at the hopes of getting at more translationally relevant data. Another approach to improve translational relevance, right, is to look at methodologies that can be used across a range of species all the way into the human clinic. And so echocardiography has been an example of that, in that we can use the same techniques, look at the same parameters across a range of species, again, all the way into the human clinic. And so as Laura mentioned, there is a lot of rapidly developing technology in this area, and so specifically, GSK wanted to take a look at this Vium technology in the hopes that we would be able to generate more accurate, reproducible, precise, and translationally relevant data. As importantly, this sort of smart housing, continuous monitoring technology allows us to allows us to potentially impact animal welfare in a way that is previously not yet done. And so, it is curious to me as a veterinarian, when we think about, say in a private practice setting, as well as for humans going into a clinic, there is always a set of data that is collected, regardless of the presenting complaint. And for a number of reasons, right? In terms of our preclinical animal models, we generally don't do that. We generally collect data in a much more focused and targeted way, in part because of the resource that it takes to generate that type of data set. And so the next couple of slides are from a IQ consortium survey, but thinking about some physiologic endpoints and how they would be useful to be able to collect in a routine and less human resource intensive way. And this would give us insight into a range of body systems, and similar type of thing in terms of behavioral endpoints as well. And again, we can do many of these things now, but the opportunity to do them in a way that takes the observer out of it and collects the data in a way that allows you to look at it as a large data set. There is, in my opinion, tremendous opportunity there. And so from my perspective, right where we are is the idea that this technology really has a lot to offer, both in terms of improving translation as well as, from the critical animal welfare side of things. So now I'm going to spend some time talking about one of the pilot studies that we did with the folks at Vium using their smart housing technology. And so just to cover the use of animals. And so the way that I'm going to talk about this is to also reference another study that our folks in the respiratory area did. And so this is a model of a lung injury using paraquat. So this is in rats, and so the rats are dosed, every 24 hours or so are placed in the plasmography chambers, and the value that is collected is called PEN-H, and essentially as that increases, that's a measure of respiratory dyspnea or distress. And so what I want to highlight here is that except for the 0.75 milligram value of paraquat, the other doses of paraquat showed changes in PEN-H at 48 hours, and then with the chart on the right, at least as measured by PEN-H, the animals are recovered by day 6. So there's the data for the 0.05 mg per kg and the 0.03. In our study with Vium, we used 0.05 mg per kg. Right. So Laura talked about the Vium smart housing and how that works. And so, right, what we did here is we collected baseline data, dosed the animals intratracheally with 0.5 mg per kg paraquat, and used some traditional measures as well as the Vium-enabled measures. And so if we look at body weight and body temperature, right, body temperature is not particularly revealing. Body weight, we see a change beginning at day 2, with maybe a hint that they are beginning to recover by day 6. Now, if we look at one of the Vium-enabled measures, right, so now we can look at the animals at night without laying hands on them. And so we pick up a change beginning 2 days after the injury in nighttime motion. And as you can see, it decreases pretty significantly, and then again by day 6 starts to recover. I do want to point out that the change in nighttime motion is about 70% compared to only about 12% in terms of body weight, right? And so this is a much more sensitive measure. It gives you a much larger window for potential compounds to show a difference. Now, when we look at a breathing rate, and so now with this Vium technology, right, we can look at the breathing rate in a continuous manner without having to put hands on the animals. You know, very different in terms of resource requirements compared to the traditional plasmography chambers. And so we see this increase in the respiratory rate, and that remains all the way through to day 6. It's also important to note that we're able to see the change as early as 12 hours after injury, and that time point actually occurs during the dark cycle when you know, people are not typically within the animal facility. If we look at the histopath from those lungs, right, right, the fact that there is very little white space or air present in the paraquat lungs at day 6 definitely fits with the fact that we're still seeing that increased respiratory rate at day 6. And it's certainly worth noting that the nighttime activity had pretty much recovered by day 6. So, you know, there are real significant benefits of looking at this model in particular within the smart housing, right? We are able to do away with the plasmography chambers and all of the challenges that come with them. We're able to get a much more in-depth, much more continuous monitoring within the animal's home cage. And the turnaround time for these studies is quicker. The other piece is that, you know, as we do more of these studies and see which measures are most sensitive, right, that allows you to then refine your study design and use fewer animals. And fewer use of animals and a more appropriate use of animals really derives many benefits, both in terms of time and cost savings, as well as things like amount of compound needed and obviously number of animals used. All right, so this is just a quick snapshot of some of the ways that, all right, we know there is a translational gap, and that smart, thoughtful use of new technology may be able to address some of that. And so one of the things that I am most excited about in terms of this technology is that now with all of these more translationally relevant measures we can get in real time, we have the opportunity to design studies in ways that certainly mimic clinical trials more, right? So we can say that rather than looking for protection from a compound in the lung injury model that we would dose, one day after the incitement of the injury, now we could say we will do that based on a change per individual animal, say on the breathing rate, right? And have more of a kind of clinical enrollment type of model. The ability to refine both protocol-related endpoints as well as general welfare endpoints increases dramatically. If you could imagine a facility that has the smart housing throughout, right? The amount of data that you can generate and then to create algorithms that really tell you, these are the animals that I need to look most closely at, right? That will give us, just huge value in terms of being able to both refine the endpoints that we use, as well as, change how we handle making the decisions about when to humanely end a study. So that was a quick view. Please, folks, if you have any questions, email them. And between Natalie, Laura, and myself, we will get back to you. And over the next year or so, I hope to be able to share some more of the work that we are doing with Vium. Thanks very much. Oh, there's my very important acknowledgements.